Good evening to all the NEET aspirants. This is your Prasanna Ma'am from NEET World. So yes, in our session today, we are going to start NCERT 12 from the chapter Sexual Reproduction of Flowering Plants. This is a very broad chapter. In a broad spectrum, so many questions have covered in the previous NEET examinations. Definitely in every year, you can expect 3 questions for sure, sometimes 4 questions also. Okay, and this chapter in connection with other chapters of your NCERT 11 can also be asked. The connected chapters for this from your NCERT 11 will be cell cycle and cell division, morphology, seed and fruit concept. These are the connecting dots where you have to refer when you are going through this chapter. And when this chapter, I told you that this is a very broad chapter. This you have to fragment it into 3 parts beta. Okay, that is pre-fertilization events, fertilization and post-fertilization events. Okay, pre-fertilization, take down parallelemma, fertilization, and post-fertilization events. Pre-fertilization, if you take, firstly we will get to know what is the reproductive organ of a angiosperm. What is the reproductive organ of the angiosperm? Flower. Okay, and the complete flower will have 4 whorls, 2 are essential and 2 are non-essential. Here in pre-fertilization events, you will get to know the complete structural description of male sex organ and the female sex organ, that is andrecium and gynoecium. Andrecium consists of what? Stamens, and gynoecium, pistil is called as synonym to it. It has what? Carpels, or you can also address it as pistil. Fine, right beta. After the structural description, you will learn about the male gametophyte and female gametophyte. What is the male gametophyte of the angiosperm? What is the male gametophyte beta? Male gametophyte is pollen grain, and female gametophyte is embryo sac. Fine, male and female gametophyte clear? Okay. So about male and female gametophyte, after which you will learn major concepts. Those are microsporogenesis and megasporogenesis. Okay, microsporogenesis and megasporogenesis. These are the concepts that you are going to finish under pre-fertilization events in sequence. Okay, now beta. After this, you will learn pollination, pollinating agents, both biotic and abiotic pollinating agents, and artificial hybridization, after which you will learn pollen pistil interaction and then double fertilization. The sequential events after pre-fertilization events, you will learn about what? Pollination, pollinating agents, okay, pollen pistil interaction, okay, pollen pistil interaction, and lastly artificial hybridization, and then double fertilization or triple fusion. This is major important segment here. Okay, after which in post fertilization events you will come across embryo development, endosperm development, seed formation and fruit formation. Embryo development, endosperm development, seed formation and fruit formation. Clear, right, ma? With this, the complete chapter will be done. After these post-fertilization events, you will learn the last two segments, which includes apomixis and polyembryony. This is the sequence that you have to go through. Okay, this is the sequence that you have to go through. From pre-fertilization events, then pollination, pollinating agents, pollen-crystal interaction, artificial hybridization, double fertilization, after then embryo development, endosperm development. First endosperm development will happen, after which embryo development will happen, whereas seed and fruit formation will take place simultaneously, and then apomixis and polyembryony. Online students, you just take a screenshot of this and you please note down now. Once it is done, then we will move on to the flower and the introductory part, how a flower is formed. Let us go to it, ma? Yes, done. So, see here first, what is the reproductive organ of the angiosperm? Flower. We know flower has how many whorls? 4 whorls, of which 2 are essential and 2 are non-essential. Non-essential whorls include what, beta? Non-essential whorls include calyx and corolla, right? Calyx and corolla. Calyx consists of sepals, corolla consists of petals, right, ma? And what are the 2 essential whorls here? 2 essential whorls. Two essential whorls include andrecium and gynoecium. Fine. Andrecium consists of stamens. Gynoecium, you can use pistil as a synonym to gynoecium. Okay. It has carpels. It can be monocarpellary or multicarpellary. Fine, right? Okay. So, this is about the flower description. The entire floral parts of the flower are situated above the thalamus, which is also known as receptacle. Clear? Now, first we will look into the male reproductive structure description. The male reproductive structure is what? Male reproductive structure is andrecium. It has filament, it has stamen, it consists of stamens. Stamen has a long tube like filament which has anther in its distal end. Okay. So, the proximal end of the filament, is attached to receptacle or thalamus. Receptacle or thalamus. Fine, ma. The distal end of the filament is attached to anther. Distal end of the filament is attached to anther, and the anther condition in most of the angiosperms is bilobed anther. Okay, bilobed, dithecous or tetrasporangiate. Bilobed, dithecous, tetrasporangiate. What does it mean? I will tell you in the structural description in the next slide. So, here in our previous slide we have learnt that the reproductive organ is flower. It has 2 essential and non-essential whorls. how a flower is formed. Okay, the decision a plant that makes to form a flower is taken much priorly than the formation of a flower. Okay, it is taken in advance only. How? First, shoot apical meristem will turn to floral meristem. How shoot apical meristem will turn to floral meristem? By reducing the internodal length, by condensation and reducing the internodal length, by condensation and decrease in internodal length. Fine. First it turns to floral meristem. This floral meristem develops or grows into inflorescence axis. Okay. This inflorescence axis will develop to floral axis, which matures to floral primordia, lastly floral bud. This floral bud will develop into a flower. Okay. This is the sequence of development which is given in the introductory paragraph of this chapter. We can expect a statement based question from this, so take the flow chart of this. First, shoot apical meristem, it will condense, decrease in the internodal length, turns to a floral meristem which grows to an inflorescence axis and then to floral axis, which then matures to floral primordia and then to floral bud, which then develops to a flower, complete flower. Fine, right ma? And next we have exercised about the male reproductive structure androecium, which consists of stamen, whose proximal end of the filament is connected to receptacle or thalamus. Distal end is attached to bilobed anther, which is also called as dithecus or tetrasporangiate. What does it mean actually? So, here if you see the transverse section of anther, transverse section, if you do transverse section of anther, the bilobed anther will be like this beta. Bilobed anther will be like this. Okay. So, this is considered as lobe 1 and this is considered as lobe 2, lobe 1 and lobe 2. Okay. In this, each lobe you have two theca, theca 1, theca 2. Here also it is theca 1 and theca 2. Okay. You have understood bilobed and dithecus, and the entire anther will have sporogenous tissues at the center. Okay, at the center it has what? Sporogenous tissues. As such, 4 sporogenous tissues are present. Okay, in each theca you have how many sporogenous tissues? 4 sporogenous tissues. Fine. That is why it is called as tetrasporangiate. Bilobed, dithecus, tetrasporangiate. Okay. And at the center, this sporogenous tissue or the 2 lobes are connected by a groove at the center. Okay. That is called as connective tissue. What is it called as ma? Connective tissue. This connective tissue is a vasculated sterile tissue. Okay. This is a vasculated sterile tissue. Fine, right? Okay. Bilobed, dithecus, tetrasporangiate, and at the center 2 lobes are connected by a connective tissue. This connective tissue you will call it as vasculated sterile tissue. Clear right now? Next. Here you see anther wall layers. Okay. Take down this. In 2 minutes we will start anther wall layers. Here we go with anther wall layers. Anther has 4 wall layers. Anther has how many wall layers? 4 wall layers, of which the first wall layer is epidermis, of course. Okay, first is epidermis. Second wall layer is endothecium. Third, it is middle layers. And fourth, it is tapetum. Fine. So here first, if you take epidermis, epidermis is parenchymatous here, it is single layered. Okay, epidermis is parenchymatous, it is single layered. In few plants like Arceuthobium, it has or it exhibits fibrous thickenings. Fibrous thickenings. Endothecium, if you see, endothecium is also single layered, it is below the epidermis. It has alpha cellulosic fibrous thickenings in its radial or tangential walls. Okay, in the tangential walls of the endothecium, which is the second layer, in the tangential walls, what does it have, beta? Alpha cellulosic fibrous thickenings. This alpha cellulosic fibrous thickenings will provide a hygroscopic nature for this. Hygroscopic nature means what? It will absorb all the moisture from the pollen grains before the dehiscence. As such, the hygroscopic nature of this tangential walls will help in the release of pollen grains. Okay, so it helps in pollen release, or you can say dehiscence of pollen grains. One more point to be added, that this alpha cellulosic fibrous thickenings will be absent in hydrophytes. These are absent in hydrophytes. Clear till here? Endothecium wall layer is clear, right? Middle layers, unlike the epidermis and endothecium, middle layers are 1 to 3 layers. Okay, and they are ephemeral. Ephemeral. Ephemeral means short lived. Okay, they are ephemeral, which means short lived. Okay, so epidermis, endothecium and middle layers, all together the major function they do is protection and dehiscence of pollen grains. Okay, all together, put together, what is the major function they are doing? Protection and dehiscence of pollen grains. Okay, ephemerals means middle layer will degenerate once the pollen grains are matured. Okay, clear with epidermis, endothecium and middle layers, beta. And the innermost wall layer is tapetum. The very first thing we need to talk about this layer is ploidy. Okay, if you have to look into the ploidy of an angiosperm, it is a diplontic plant, right? Okay, the ploidy will be diploid, flower will be diploid, essential, non-essential whorls, ploidy will be diploid. As such, the anther ploidy is again diploid, right? Epidermis diploid, endothecium diploid, middle layers diploid. But then tapetum ploidy is polyploid condition. Okay, the major function of these 3 layers is what? Protection and dehiscence of pollen grains, but the function of tapetum is nutritive. It provides nutrition to sporogenous tissue. Fine. Fine, it provides nutrition to the sporogenous tissue. So, the tapetum is the innermost wall layer which is surrounding the sporogenous tissue. The main activity it is doing is providing nourishment to the sporogenous tissue. It also provides out ubish bodies, it synthesizes ubish bodies and sporopollenin, which is the exine wall layer of the pollen grains. Okay? Sporopollenin, which is the exine wall layer of the pollen grains. This is the complete description of the 4 wall layers of the anther. And I have already told you about the connective tissue, which is present at the center connecting 2 lobes of the anther, which is a sterile vasculated tissue. Okay. Clear till here, ma? Any doubts you are having? No, right? Clear with the anther wall layers? Now, here at the center I told you, you have sporogenous tissue, right? So, at the center here at the sporogenous tissue, you have spore mother cells or pollen mother cells. Clear about wall layers? Now, we are talking about sporogenous tissue. These 4 sporogenous tissues are there, right? At the center they have what cells? Spore mother cells, you will also call it as pollen mother cells. So, sporogenous tissue will have spore mother cells or pollen mother cells. Their ploidy is what, ma? Diploid. They will undergo a meiotic division or a reductional division. Okay. Before undergoing division, one point should be clear, beta. Each spore mother cell is covered by a callose wall layer. Okay, each spore mother cell is covered by a callose wall layer, and each spore mother cell after reductional division, that is after meiosis 1 and meiosis 2. How many daughter cells are formed? 4 daughter cells are formed. Those 4 are haploid microspores. 4 microspores which give tetrad condition, they are haploid. Fine, tetrad condition, haploid. Fine, right, beta? Okay, 4 microspores which are haploid provided tetrad condition. All these 4 microspores are present inside the callose wall layer only. Once the microspores are mature to pollen grains, then callose wall layer is degenerated by the enzyme callase. The wall layer callose is degenerated by callase and microspores will be released out as matured pollen grains. Microspores will be matured to pollen grains. Pollen grains are what? Male gametophytes. Inside pollen grains only male gametes will be there. I already told you what is the meaning of a gametophyte when I am talking in plant kingdom. What is a gametophyte? Any body or any plant body which is bearing the male gamete or gamete is called as gametophyte. Male gametophyte means it is bearing male gamete. Okay. So, pollen grain here is the male gametophyte. Clear, right? How spore mothers inside sporogenous tissue you have spore mother cell or pollen mother cell, and this undergoes reductional division meiosis 1 and 2, and then it forms 4 haploid microspores. 4 haploid microspores are released out in what condition, beta? Haploid condition, and callase will degenerate callose wall layer, and this microspores will be matured to pollen grains, which is the male gametophyte. Now, we will look into the structural description of a pollen grain. How a pollen grain structure will be there? Pollen grain structure, first of all, the wall layer of the pollen. Okay, pollen grain wall is made up of sporoderm. Sporoderm. Fine. And it has 2 wall layers. Outer wall layer is exine, inner wall layer is entine. Okay, 2 wall layers. Outer wall layer is exine, this is outer, and inner wall layer is entine. Outer exine wall layer is made up of major component, major toughest organic compound which is known. Okay, that is sporopollenin. okay, which is sporopollenin. Fine. Most toughest organic compound that is known. Okay. Because of the sporopollenin only, the pollen grain is persistent for many years of time. The sporopollenin is undegradable by any acidic condition or alkali condition or any high temperature condition. This is resistant for everything. Okay. This sporopollenin in the exine wall layer, it is not completely present on the exine wall layer. At some regions it is absent beta. Those regions are called as apertures or germ pore region. From this germ pore only, whenever pollen grain lands onto the suitable stigma, from the germ pore region only pollen tube will grow. Okay. So, here it is clear, right? Sporopollenin which is present in the exine wall layer, it has what? In the germ pore region, sporopollenin is absent. Next is inner wall layer, that is intine. Intine is made up of cellulose and pectin. Cellulose and pectin. Fine. Now, we will see in how many celled condition the pollen grain is released out. Mostly the pollen grain is released out in two celled condition. Okay. That is, it has a vegetative cell and a generative cell. Pollen grain will have a vegetative cell vegetative cell and generative cell. What does it mean? Even before the release of pollen grain, okay, at the time of release, inside the pollen grain, one mitotic division will happen. Okay, one mitotic division will form two cells, right, two daughter cells. One is vegetative cell and the other is generative cell. In the vegetative cell consists of a large vacuole, okay, large vacuole and irregular shaped nucleus. Irregular shaped nucleus. This is visible, no? Yeah. And the generative cell will contain spindle shaped nucleus. Here you can see it consists of spindle shaped nucleus. Clear, right? Structure of pollen grain is clear, no ma? In 60% of angiosperms, in how many celled stage the pollen grain is dispersed? Two celled stage. One is vegetative cell and the other is generative cell. So, pollen grain will undergo how many mitotic divisions? One mitotic divisions. Pollen grain wall layer is made up of sporodon. Outer wall layer is made up of exine. Exine consists of sporopollenin. In which regions of exine sporopollenin is absent? Germ pore regions or aperture regions. Next to exine you have what? Intine. Intine consists of cellulose and pectin. Clear, right? Now, we have to go through, this is pollen pistil interaction, I will explain you there more clearly. 4 segments you have to cover with pollen grains. Okay. Take down. First is pollen allergy. Second is pollen products. Third is pollen viability. Fourth is pollen banks. Okay. So, pollen allergy, if you see, pollen allergy, few plants, actually the pollen whichever is released, few people who are very sensitive to the pollen grains or pollen dust. Usually if you go for walking also, those people who are sensible for the pollen allergy, they will get skin rashes and all. Even those people who have some lung disorders and all, they are very sensitive to the pollen grains. But in our NCERT, 3 plants are mentioned where we have to remember it for sure. That is Parthenium, that is carrot grass, Parthenium, which is commonly called as carrot grass. Fine. and Amaranthus and Chenopodium. These plants' pollen grains, okay, they cause severe bronchial afflictions and allergy. Bronchial afflictions. Next, pollen products. It is believed that most of the pollen products or pollen consumption have increased the capabilities or abilities in the athletes and horse races and all. So, that is why pollen consumption is mostly taken by the athletes, okay, which are highly nutritive. Pollen consumption by athletes. And next, pollen viability. What does the term means viability? Pollen viability. The time period from that it can germinate on the stigma, that time period is called as pollen viability. Okay, that time period is variable for variable pollen grains, or it is variable for different families. It can be just 30 minutes, pollen viability can be 30 minutes, like some cereals in rice and maize, in some cereals like rice and maize, it is just 30 minutes. Rice and wheat, it is just 30 minutes. It can be for 3 months, for few months, okay, in Rosaceae, Leguminosae and Solanaceae. Okay. Pollen viability can extend up to months in Rosaceae, Leguminosae and Solanaceae. Lastly, pollen banks. We can preserve pollen grains for years together by following a method called as cryopreservation. Okay, cryopreservation. What is cryopreservation, beta? That preserving pollen grains at -196 degrees Celsius in liquid nitrogen. Okay, preserving pollen grains at -196 degrees Celsius in liquid nitrogen. Clear, no? Fine. Now, female gametophyte and structure of ovule, and then I will leave you, okay. Just 10 or 15 more minutes, I will leave you. This you already know. What is the female reproductive structure? Not gametophyte. Gametophyte is embryo sac. Female reproductive structure is gynoecium, which is also known as pistil. It can be monocarpellary or multicarpellary. Single carpel will have single unit of stigma, style and ovary. Stigma is the receptive portion of pollen grain, and this long tube will carry the pollen grain, okay, to the embryo sac, that is female gametophyte, okay, style, and the basal bulged portion is what? Ovary. Fine. So, here monocarpellary, it has single carpel unit. Multicarpellary, it has many carpel units. It can be apocarpus or syncarpus. Apocarpus where pistils are free, carpels are free from each other. Syncarpals where carpels are fused with each other. Okay. So, apocarpus you can take the examples of lotus, rose and michaelia. Syncarpus you can take the examples of mustard, tomato, hibiscus and papaver. Clear? Clear till here? Monocarpellary and multicarpellary condition. This is the diagram that is given in your NCERT for michaelia, which shows apocarpus condition. This is the syncarpus condition of papaver and hibiscus. Here, so here you can see stigma, style and ovary in a single carpel. Inside ovary, ovules were placed. Okay, ovules were placed. The arrangement of ovules inside the ovary is called as placentation. Once let us quickly recall the placentations. We have 5 types of placentations, right? What are they? Marginal, axial, parietal, free central and basal. Very good, Abdul. Okay, so marginal placentation examples include pea and bean, Fabaceae family. Okay, axial placentation, China rose, tomato and very good. Parietal placentation, you can say mustard, argemone, good. Next, free central, dianthus, primrose, dianthus and primrose. Okay. Next you have basal placentation, which is seen mostly in the single ovulated plants. Okay. So basal placentation you see in sunflower. Okay. So these are the examples, just a quick revision of placentation. Now you see the structure of ovule. Okay, very important. Tomorrow we will see types of the ovules. So here you see my structure of ovule. If you see, most of the ovules in the angiosperm, it is anatropous ovule. Mostly in the angiosperms, what type of ovule you will have? Take down, anatropous ovule, where ovule is inverted to 180 degrees. Ovule inverted to 180 degrees. Here if you see, this is called as stalk of the ovule. Okay. How ovule is attached to the ovary, like how we are attached, fetus is attached to the womb, right, through the umbilical cord in human body. Same the case, the ovule in plants is attached to the ovary by a stalk called as funicle. Okay. So this funicle is what? Stalk which attaches ovule inside ovary. Next, this is small structure which is the connection between funicle and the ovule. Hilum connects funicle with ovule. Okay, and ovule is covered or encoated by integuments. Integuments post fertilization will mature to seed coat. As ovule post fertilization turns to seed, right, integuments will mature to seed coat. And here the small pore like structure, which is present is called as micropyle. In anatropous ovule, the micropylar region is in close association with hilum. Micropyle is closely associated with hilum. Opposite to the micropyle, the basal portion is called as chalaza, where antipodals are present. Okay. And here the cells which are present inside the ovule are called as nucellus cells. Important. Okay. The ploidy of ovule is what? Diploid. Mostly the ploidy of nucellus cells will be asked. The ploidy of nucellus cell will be diploid. Fine, right? This is the structural description of ovule. Do you have any doubt over here? No? Okay. In a single minute, we will revise what all we have discussed today. We have started sexual reproduction of flowering plants, complete concepts inside that, pre-fertilization, fertilization, post-fertilization events, flower description, how a flower is formed, okay, and then male reproductive structure. What is the male reproductive structure? Andrecium consists of stamen, structural description of stamen, filament and a bilobed anther, bilobe dithecous tetrasporangiate. And then we have looked into the microsporogenesis, after which we have learnt structure of pollen grain, pollen products, pollen allergy, pollen viability, pollen banks, after which we have looked into female reproductive structure, okay, and in female reproductive structure Gynoecium is also addressed as pistil. It can be monocarpellary or multicarpellary. Multicarpellary, it can be Apocarpus or Syncarpus. Apocarpus example, Michaelia, Syncarpus, papaver and hibiscus. Okay. And then we have looked into the structure of ovule. Clear everything? Tomorrow we will look into the types of the ovule, female gametophyte, pollination, okay, this artificial hybridization, pollen pistil interaction, double fertilization. These events we will cover in the next class. Okay. So, that is all for today's session beta. Thank you.